Hi guys, this is Preeti and you're watching Meta Solution. In this video, I'll be teaching you on the RS question. That is transportation across the cell membrane. I hope all of you are doing well, and then your internals you are going through or some exams, and you will be scared of a university exam. So if you go through all RS questions, it will be very helpful for you to get good marks in the university exam. And in this video, I'll be teaching about the transportation across the cell membrane, which is important in perspective of both biochem and then physiology. For transportation across the cell membrane, cell membrane is a bilipid layer. So it hydrophilic part and hydrophobic part which is hydrocarbon chain, and hydrophilic part which is water soluble. So it allows aqueous medium till here, and it cannot allow because this is a hydrocarbon chain are lipid soluble. They are lipids, so like dissolved so lipids, only lipids can pass through this, like water soluble or aqueous or polar substances cannot cross this membrane. So they require some carrier proteins, and we'll discuss the types of these transport across the cell membrane. Passive transport means without energy, like from a higher concentration to the lower concentration, from the that is downhill transport is a passive transport. In passive transport, we come across different examples. Simple diffusion is also like a type of passive transport. So it doesn't require any energy. It water or solvent moves from a higher concentration to the lower concentration, and without the help of proteins. Simple diffusion and facilitated diffusion are one in the same, with the help of in facilitated diffusion, with the help of protein channels, the particles move, this attains saturation. When all protein channels are filled, it attains saturation, whereas in some simple diffusion, it is independent of protein or like a living system. This depends on protein channels, so this is a living system. It depends on living system. So it doesn't attain saturation because simple diffusion does not depends upon the protein channels, but this attains saturation. And osmosis is also a passive transport, so it is a type of passive transport, so it doesn't require any energy. Osmosis is a mainly water movement. Water moves from the higher concentration to the lower concentration across the semi-permeable membrane. This is known as osmosis. Filtration is nothing but normal filtration. If we filtrate the main thing, upper part, but retained, and there will be retained and filtrate. And the bulk flow due against the gravity, like a large number of molecules are pulled. This is known as solvent drag. Solvent drag means when there are more number of particles, and then more number of particles, secondary to that, the solvent is pulled, that is known as a solvent drag. And then we come across active transport. If we go through active transport requires energy, and it moves, particles moves from lower concentration to higher concentration. So this is also known as uphill transport. It requires energy. These are protein dependent, and there are two types of active transport: primary active transport and secondary active transport. Primary active transport directly depend upon the ATP. For the uphill transport, that is for movement of molecules from lower concentration to higher concentration, whereas in secondary transport, one is utilizing primarily one, it is you take, this is a membrane. So sodium is utilizing ATP and then moving sodium. With the help of these the concentration created by these sodium will helps in transportation of other things. So this is our secondary active transport. The indirectly it utilizes the ATP. Secondary to one, the primary transport, the transportation takes place. This is known as secondary active transport. And there are vesicular transport also. Vesicular transport, there are two types: exocytosis and endocytosis. It includes involves the formation of vesicles, and then the release. Exocytosis example will be neurotransmitter, and endocytosis the example will be phagocytosis. During phagocytosis, endovesicles will be formed, and then release the proteolytic enzyme from the lysosomes, and then they degrade it. And epithelial, across the epithelia, these are, if we take, these are epithelial cells. If they transport across these epithelial cells, we call as a transcellular transport. If the transportation is in between the two epithelial cells, that is in between interstitial epithelial cells, we call as paracellular transport. And these are the brief explanation about the transportation across cells, and you can write these differences and also to gain more marks. You'll get more marks if you write like this, like paragraphs, not like paragraph differences and then flow charts. You will get more marks. So I thought of I explaining this also. Simple diffusion and facilitated diffusion. Mode of diffusion: simple diffusion does not require any carrier protein, whereas facilitated diffusion requires carrier protein, because since it is require carrier proteins, it doesn't require carrier protein, so it does not attain saturation, whereas this requires carrier protein. If all the carrier proteins are failed, then it attains saturation. And then competitive inhibition is not present in simple diffusion, as it doesn't depend upon any carrier proteins. Facilitated diffusion depends on carrier protein, because same carrier protein will be will be dependent by other substances. So rate of diffusion in simple diffusion will be slow, as they have to cross through the bilipid membrane. So in facilitated diffusion it will be faster. Specificity, it has no specificity because it doesn't depend on any carrier protein. They are specific to their carrier proteins. So in facilitated diffusion the specific proteins are required. And again, in simple diffusion we can speak about lipid soluble and lipid insoluble. Lipid soluble can easily pass through the membrane. Example, oxygen, urea, so they directly diffuses across this membrane. Whereas water insoluble, like ions, cannot move across the lipid ions. Like ions means they are absolutely water soluble, so they cannot cross the bilipid layer. They depends upon voltage gated channels or ligand gated channels. Voltage gated channels mainly depend upon the potential difference across the two membrane, like outer membrane and inner membrane potential difference. Ligand gated depends upon the chemicals. And if we discuss about the factors that on which the simple diffusion depends, means like a molecular size. If small molecular size, it easily passes through the membrane. If it is a less molecular size, it easily passes through. If it is large molecular size, like molecular weight or size, it is difficult. You can think that a thin person can run faster than a person with a heavy person, but than a heavy person. So simple diffusion mainly depends upon the size of the particles. And then lipid permeability, like permeability of the membrane, that is lipid solubility. If it is more lipid soluble, if it is a lipid soluble, then it can easily pass through the membrane. If it is water soluble, it cannot be passed. And then if we come up with the thickness of the membrane, if it is a thinner membrane, it can easily pass through. If it is a thicker membrane, we cannot penetrate through it and then pass through. So this and it also depends upon the temperature. If it is more temperature, it can easily pass through them. If it is less temperature, it cannot pass. And it also depends upon the concentration gradient, pressure gradient. Concentration gradient, if the difference is more, so easily can from one compartment to it can pass through the other compartment. If you like this video, or if you think this video is helpful for you, please stay tuned to my videos. Thank you.